It's great to see some former patients, current patients, and I get to talk about PMP and appendix cancer to obviously to my own patients, also to physician forums and conferences, some of the ones that Jerry mentioned. But it's really an honor to speak with this group today. I'm really honored. So thank you so much for inviting me. The first slide here, I'll just give a brief disclaimer. So I am not a geneticist. I'm a surgeon. I don't do basic research either. I do some clinical research, so patient-based research, involved in some clinical trials and sort of database kind of research, statistical research. But I'm certainly not a geneticist and not a basic scientist. But hopefully, I'll still be able to give you kind of a little bit of background of genomics and some of the projects that we've done. And I say we, and I'm being very sort of overstating a little bit. Dr. Lowy is actually my senior partner at UCSD, as many of you know. And he and his collaborators did most of the work that I'm going to be presenting today. And they presented some of this research at we had a PMP patient talk last fall, and I was asked to present some of that data. So that's what I'm going to present. So I didn't do this the research here. The slides mostly come from me. But anyway, without that disclaimer, I just thought I'd talk about the genome to start with and a little science this morning. But like I said, I'm not going to delve too deep in this. But I think it's important to understand. I think obviously many in this room, we all come from different places. Some of you have not heard much about DNA and genome. Some of you are world experts in it. So I'll start with the basics. Really, the genome is just the set of your DNA. It's just the entire set of an individual's genetic material. And the DNA, is housed in each cell, and each cell basically has an identical copy of your entire genome. And there are a few mutations, but for the most part, the cell in your eye has the same DNA as the cells in your liver, as in your skin, as in your pancreas, in your colon. They all have the same genetic material. And that genetic material is housed in chromosomes. There are 23 pairs of chromosomes. They look like there's these X things here. And basically the DNA is all wound up very, 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 very tightly into these chromosomes. And then it's bound by these proteins, nucleosomes, and histones. If you actually took the DNA from a single cell and stretched it all out, it would actually be about 2 meters, so a little over 6 feet in length from one cell. But each of your cell has that equivalent amount of DNA. And so if you look at the basic structure of DNA, we talk about this helix, this alpha helix. It's sort of like a ladder that's kind of twisted on itself, and then there's these rungs of the ladder here, and those are, many people can't see that I'm sure, but those are what we call the base pairs. And that's the basic unit of DNA, is these individual base pairs. And there's actually only 4 of them. So our entire instructions for life and all of our cells are written with a 4-letter alphabet, which is pretty amazing actually. And so that's what these A, G, C, and T letters are designated. They're pairs, so they always bind to each other on the opposite ladder, on the opposite rung of the ladder. So A's always bind to T's and C's always bind to G's. So if you know the sequence or the order of those letters on one strand of DNA, you know what the opposite strand will be because they always have that binding pattern. So that sequence, the order of those letters, A, G, C, T, maybe G, 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 C, C, T, A, A, that sequence over the whole 3 billion base pairs that make up your genome is basically, that's the instructions for your life. That's the instructions for each cell. And within your whole length of DNA, there's actually only about 20,000 genes. Sounds like a lot, and it is a lot. It's actually about twice the amount of a fruit fly. But it's less than corn. So, yeah, that's true. Yeah, that's right. So it's actually only about 1% of your genome makes genes. So what is a gene? My 5-year-old son saw me putting together these slides, and he said, "So my cells have genes?" Like he's my and I said, "Well, yeah, sort of." So a gene is basically the part of the DNA that makes a protein, and a protein is basically how the DNA carries out its instructions. So the DNA, I'm not going to go into this, makes RNA, which ultimately leads to proteins. So only about 1% of your whole genome actually makes protein. Well, what does the other 99% do? For a long time, it was called junk DNA. It turned out it's not junk at all. It actually, it's very important to help regulate the other parts of the DNA and help it fold in some ways. So it's actually interesting that only a very small fraction of the genome actually makes the proteins which are what carry out the functions. And it turns out everybody in this room, look at your neighbor, you only have about a 0.1% difference in your genetic materials from every other individual. So 99.9% of our genetic material is all the same. So there's very subtle differences that obviously make very big differences in how we look and behave and act and live. So that's a brief history of that. So cancer, you can really think of it at its core as a disease of the genome. So cancer really is just an accumulation of mutations of the cell's DNA. And you can see here in this upper left part of the screen, there's a normal cell. I think these are in your handouts too, so you can see. A normal cell lives its life in your body, it divides and has daughter cells, or son cells, I guess. And those cells behave and do what that cell is directed to do by its own DNA. Well, over time, as a result of environmental factors, as a result of maybe underlying genetic conditions, those cells can acquire mutations where there's an alteration of its own DNA. Maybe one mutation doesn't have any effect on that cell, but over time, cells can acquire additional mutations. And at some point, given enough mutations in the right locations, an individual cell can become a cancer cell. And that's what this shows here. And this sequence of mutations has been worked out in many cancers. For example, colorectal cancer, very well known and well described mutations that colorectal cancer has. It's listed here at the bottom. So you start out with the normal epithelium, the normal lining of the colon. It acquires a mutation in a gene called APC, and then that causes a normal epithelial cell to become a polyp, basically, and that's how the polyp is formed. And then if that polyp obtains additional mutations in KRAS, or in DCC, or in P53, it can become a full-blown colon cancer cell. And so that's very well worked out in colon cancer. This is not how it happens in every case of colon cancer, but this is the vast majority of the time. And this has been described in pancreas cancer and melanoma and other cancers, but obviously PMP and appendix cancer is quite rare, and so it's not been well described so far. So the kind of mutations you get are listed over here. It's kind of a lot of big words, but the basic point, I think, is that you can have all kinds of mutations that do different things in your DNA that affect how that piece of DNA gives instructions and how it affects that cell. So this is a SNP, we call it, a single nucleotide polymorphism. All that is just one of those base pairs is mutated. So it went from a G to a C or a T to an A. And that may have no effect on that cell. It may have no effect on a gene, even if it's within that gene. But it can. Insertion, you can have a small little yellow piece of DNA in this picture that gets inserted where there shouldn't be a G. There's an additional G added. There can be a deletion. And then you can have structural variations. The whole length of DNA, there can be some chunks of it that are switched. That's called an inversion or translocation. You can have whole segments that are deleted, whole segments that are inserted. And then aneuploidy here is just a term that means there can be copy number alterations. When a cell divides its chromosomes, and then each daughter gets half of that DNA. Well, sometimes for whatever reason, one daughter cell will get an extra chromosome or an extra copy of DNA, and that can have ramifications on that cell. And so there's all kinds of ways that mutations can be acquired. But ultimately, those mutations all lead to a cell behaving like a cancer cell. And I realize this is really small print, but basically there are certain hallmarks of a cancer cell that we describe. And these hallmarks are what make cancer cells behave like they do. And they have these hallmarks include things like evading the immune destruction. The immune system probably would eradicate a lot of cancers, but somehow cancer cells have acquired mutations that allow them to avoid being eradicated by the immune system. They have replicative immortality. The cells just continue to divide. They don't respond to the normal inhibitory factors that suppress growth, so that's why tumors continue to grow. Tumor-promoting inflammation, invasion of metastases, angiogenesis, they grow new blood vessels, and so there's obviously therapies against that. And so there's all these hallmarks of a cancer cell that we see in a cancer cell, how it behaves. Well, as we understand the mutations of the cell that lead to these hallmarks, various targeted therapies have been developed to target each one of these hallmarks of a cancer cell. So if we know in a colon cancer, for example, what mutation led to one of these hallmarks, then we can develop a drug that may target that mutation. And so that's been true with angiogenesis inhibitors, for example. Avastin, we heard about this morning over there. That's a drug that targets new blood vessel growth. And there are many, many other ongoing targeted therapies that deal with these mutations. Well, the problem is, in PMP, you don't really know what these mutations are, and so that's kind of what has instigated the genomics studies that Dr. Lillian did at UCSD. So, just one slide on cancer genomics. We have someone here from Illumina. I can talk about this more than I can, but the technique of genomics is a relatively new field. We didn't have ability to sequence this genome because it's such an incredibly huge genome. And to discern all the sequence of the entire genome has not really been available until recently. So the Human Genome Project started back in 1990, and at the time it cost about $10 per base pair. We have 3 billion base pairs, so you can imagine what the cost would have been if that cost had stayed the same. Turns out they were able to sequence the entire human genome of one human. It was actually a collection of people, but essentially they sequenced an entire genome, all 3 billion base pairs, after 13 years and at a cost of $2.7 billion. They actually had budgeted for $3 billion in 15 years, so it's one of the few government projects that was cheaper than they asked for. But you can compare it to now with Illumina and other companies that can do this for maybe $1,000 to $1,500 and ultimately in a few days. The world record, I think, is about 26 hours. I think a child had their genome sequenced. This is over one day. It took 15 years, essentially, for the first genome to be sequenced. It's incredible. This slide just shows how the ability to sequence DNA has outpaced even our computational rate over time. Moore's law is a computer science term that says you have a doubling in computational power every 2 years. So if you apply that to DNA sequencing, you would see that white line, but actually DNA sequencing has far outpaced our ability to what we would expect from computers. So in cancer, why do we care about the sequence? Well, it's because if we can identify what mutations exist in a cancer cell, then we can have a better understanding of that cancer. It can also allow us to get targeted therapy, like I talked about in a couple slides. And then there's also screening, diagnosis, and surveillance, because if we can understand what mutations will lead to what cancers, and we can test for those mutations, we might be able to screen people that before they develop a full-blown cancer. Diagnosis, we can diagnose cancers if we can potentially identify what mutations are always present in a certain cancer. And then surveillance, perhaps we can check for recurrence of mutations down the line in some way. So there's a lot of advantages and a lot of downstream effects of cancer genomics. Well, the problem is in PMP, none of this was known. So no one really knew what mutations occurred in someone that had appendix cancer. And so that's why Dr. Lowy and collaborators at UCSD and others around the world really have started to look at this. And so basically the goals were just to understand a little bit more of the molecular biology of PMP, also to identify therapeutic targets, and then also to suggest possible clinical trials depending on what mutations were found, and then to direct research efforts. So, I just have a couple slides on appendix cancers. We all are very, very familiar with appendix cancer, but I think it's important because the terms are very confusing, and even there's a lot of controversy within the pathology world and within the surgery world about what kind of appendix cancer you have and what is PMP exactly. This is kind of, as a surgeon, we have a simplistic mentality of things a lot of times, but I viewed it to high grade and low grade, and I think our pathologists by and large agree with this demarcation. So we know there are low grade varieties of appendix cancer. Now the preferred pathologic term is lamin, per the WHO criteria, and these are low grade appendiceal mucinous neoplasms. But basically, these are the typical PMP picture. This is the type of, it's a proliferation of epithelial cells that really under the microscope look pretty bland. They don't have a real appearance of cancer, but they produce mucin. They can push through the wall of the appendix and can disseminate. And then there are high-grade appendix cancers that look, for all intents and purposes, like an adenocarcinoma, which is the type of cancer that you might also get from your colon or your breast or somewhere else. And so they have all the features. The slides on the right are what they look like under the microscope, but they look like a cancer cell. And so they invade through the wall, they cause all kinds of other problems. And so for today's talk, and also just really how we do this, we divide this disease up into low-grade and high-grade. And some of you are familiar with that dichotomy, but that's kind of how we talk about it. And so that's kind of the terminology that I use this morning. So how do appendix cancers spread? We don't really know, is the bottom line. There's not like a real good animal model that we can use that has PMP that we can follow. So it makes it difficult to study. But we know that there's at some point a tumor, a neoplasm we call it, that lives in the appendix that makes mucin. And then somehow that mucin gets ruptured or spread into the peritoneal cavity. It could just be that the appendix ruptures, like any appendicitis can do. The mucin causes it to pop like a balloon. And some of these red tumor cells then leak out into the peritoneal cavity and then they grow. Mucin can extrude through the wall of the appendix. Maybe it's not a full-blown rupture, but the mucin just gets pushed out. Or maybe there's actually invasion of the cells into the peritoneal cavity. But whatever happens, it all leads to the same endpoint, which is basically the tumor of the appendix ends up disseminating through the peritoneal cavity and leading to PMP, as we call it. So that's kind of the nuts and bolts. The problem is, particularly in low-grade disease, if you really zoom in on the tumors that are in the peritoneum, this is what we see. And this may not mean much to most people, but if you're a pathologist, if you're trying to do studies on these cells, it's very difficult because all of this stuff over here, all this kind of dark or light gray stuff and this pink stuff, is mostly acellular material. It's mostly not cells. It's actually mostly mucin. And that's a challenge, as some of you know, on pathology reports, you see that there's often just a lot of mucin, and there's not a lot of cells that make the mucin in the tumors. And that makes it tough to do studies on those tumors because you have to find those cells in order to test them. And so that's a big challenge in doing research on this disease. And you can see here, this is all connective tissue. So there's a few cells scattered here, but they're not even tumor cells. They're stromal cells. They're cells that surround the tumor. There's this thin strip here is actually the epithelial cells. These are the tumor cells. And this is often what it looks like. And so to study the tumor cells that are creating PMP, we have to be able to find a way to cut out those cells and just look at those cells. And there's a technique called laser capture. Yeah?
Is there chemical markers?
There are, there are. Yeah, so there are markers that they can do to stain the mucin. Usually they don't need to do special markers because you can see the mucin it looks different than the cells themselves.
Is the PMP mucin different from other mucin?
It is, yeah. It has some, it's not a lot to know about it, first of all, but yeah, there seems to be some differences. Yeah. Like the mucus, the mucin that your body normally makes in your GI tract or in your respiratory tract, it does look a little different, yeah. So, the project that we did was basically to look at the DNA of those tumor cells and detect what kind of mutations were present. And so this was done in two phases, as lots of these kind of studies are done. The first phase that Dr. Lloyd and collaborators did was the discovery phase. So they basically took tumors from 10 patients, 9 of which had low-grade disease, 1 of which had high-grade disease. And they did laser capture dissection. That just means to cut out with a laser the individual cells and test the DNA of those cells, as opposed to all the other stuff around it. They did whole exome sequencing, which is basically, exome is the part of the DNA that codes for proteins. So they only really looked at that 1% of the DNA that makes genes. There could be mutations in the cells in that other 99%, but this was just the first approach to this. And then they looked at copy number alterations and mutations within that cancer cell. And then once they identified what genes were commonly mutated in that group of 10, then they went to another 19 patients and were able to look at those particular mutations. Now that we knew what mutations were commonly present, you could look in a bigger set of patients and see if those mutations were present in that second set, and that's called the validations. And there were different techniques used, but I don't need to go into the details there. So this is kind of really the findings in one slide of what the project was. And so you can see there's a whole list here of different genes that were mutated. And there were actually about over 2,000 mutations found among that first set of 10 patients. And there were in at least 2 or more patients, there were as many as 25 genes mutated. Some of these had cancer related based on what that gene does and how it was mutated in other cancers. So that you can see all the blue are those 25 common mutations that was found in those cancers. The red here are non-significant mutations that were found. And then there were, many of these have cancer relationships in other diseases, and so they were found. And you can see the top 2 ones at the top 2 mutations that were found in this disease was in a gene called KRAS and a gene called GNAS. And so those were by far and away the most common mutation. The other interesting thing is, unlike colorectal cancer, mutations were not commonly found in a gene called APC. Remember that first slide I showed, or second slide I showed, the APC mutation is the initiating mutation for colon cancer in most cases. That wasn't even found in this disease. And nor was P53 or TP53, commonly mutated gene in many, many cancers, very rarely mutated, especially in low-grade patients. And so it seems from the outset already that this is not like colon cancer, which is no surprise really to anyone here, but that's a big surprise actually to a lot of people that don't see it, that don't see PMP or appendix cancer. It's lumped with colon cancer actually in a lot of ways. But genetically it seems to be quite different, and obviously its behavior is quite different too.
Dr. Baumgartner?
Yeah.
So how much volume of specimen do you need to do this? Because my understanding is it's kind of hard, and particularly because there's usually a lot of inflammation as well.
Right.
And also my second question is, when you bought the UCSD, is that considered a contaminant? Does that make it even more difficult?
It does, yeah. I don't know how much they actually needed for the first part of this study, but it's pretty sizable amounts of tissue. Now, most of our patients have a fair amount of tissue that we save, a tumor that we save, but you're right. And then the mucin for this study was kind of a contaminant because that was not something that we wanted to test. Now, when you have a test by Foundation Medicine or one of the other companies that can do molecular profiling, you'll hear about, it's a little different. The techniques that were done were a little different than this first big sequencing method. And so they can they can basically test for certain genes that we know are going to be present, and so you don't need as much tissue.
So could one theoretically request a surgery just to get?
Well, right. So we get that question a lot. Yeah, so to do molecular profiling, now that we know what mutations are common, we can look for certain mutations or look at a smaller subset. Remember, when we did the sequence, we're just looking at the actual sequence. But we can do Foundation or other companies with 400 plus genes. And, can we do surgery for that? We don't often do that. Sometimes you can do it with just a simple biopsy, but often it depends on how cellular it is and how big of a sample. I mean, that's what you're getting at.
Yeah.
I would say in general, when I get that question, I wouldn't recommend doing an invasive surgery for that reason, unless there's really no other option that we have. But a lot of times it's a side benefit. Well, there's some symptoms that we want to treat, even though we don't think we can do HIPEC anymore. Maybe we can go in and do a debulking and get a bunch of tumor to do this kind of testing too.
So in conjunction.
In conjunction.
Palliative.
Exactly.
Yeah.
Right. That's something I would.
I think that's been the real difficulty for me, is adequate specimen or tissue.
Exactly.
Size. And then when you have inflammatory cells, you can't tell sometimes when
Exactly.
positive as well.
But I would say that slide I showed with Moore's law and all that, the threshold is changing all the time. How many cells we can use to detect DNA. I mean, we may be at the point down the road that we just need a couple cells, and we can do that. Now, there's a whole other issues of heterogeneity that I'm not going to talk about today. But maybe the tumors in one part of your abdomen look different than the tumors in the other part, and does that matter? Or what's the real genome of the cancer? It's not really unknown.
Thank you.
Yeah.
You say there were 2173 mutations and mutations. Is
Right.
that typical of a cancer patient, or is that a higher number of mutations than you normally
so
find?
it's pretty typical. I would say it was surprising to us because we thought it might be much lower. Because this disease is so slow growing in general that we think that it may not have as many mutations. But I would say that number is hard to predict. There are, for example, melanoma, for whatever reason, has lots and lots and lots of mutations. Other cancers that are at least as aggressive, if not more, pancreas cancer and others may have slightly less. So it doesn't always correlate the number of mutations to how aggressive the disease is, but in general, there's kind of a relationship there. And so we thought we may not detect as many mutations as was found. And so, yeah, so maybe, you know, it depends how you classify mutations and how we found them. Remember, we just looked at the coding sequence of the DNA, only that 1% of the DNA. There's likely lots of other mutations, but they may not be driver mutations, as we call it. They just may be a mutation that you have in that cancer cell that didn't have any influence on whether it became a cancer cell or not. And so, but the fact that there were 25, at least in most patients, was interesting to us.
Dr. Baumgartner?
Yeah.
On the previous cell, is there a difference in between the high grade and low grade?
Yeah, so I'm going to talk.
Relative to the
Yeah, so that's exactly right. Perfect segue there. So we focus then on the KRAS and the GNAS mutations, because they're far and away the most common two mutations in this disease. And you can see here we basically looked at by low grade and high grade for each GNAS and KRAS. And so the validation set is listed here as well as a discovery set. So that whole 29 patients are lumped together in this group. And you can see, so the red bars, or red colored bars, are patients that had mutations, and the blue ones are where there was no mutation, or the wild type, as we said, which means the kind found in nature without a mutation, so a non-mutated gene. So if you look at, for example, low-grade patients, there were 23 total in our entire set. Almost all of them had KRAS mutations. In fact, I think it was like 80–90% had KRAS mutations. So virtually all patients in low-grade disease had KRAS mutations. But also, we found a similar result in the high-grade patients. So the vast majority, there were 6 high-grade patients in our whole group. Most of them had KRAS mutations as well. So both low-grade and high-grade seem to have KRAS mutations. However, we didn't find that when we looked at GNAS. GNAS is another gene. Almost all the low-grade patients had a mutation in GNAS, but hardly any of the high-grade patients had a mutation in GNAS. And so what is that?
Joel, thank you
Oh,
for
no problem.